Welcome to Mr. Chalk's Revision Tips.
When answering exam questions, there are a few key things to remember, right? So just remember, exam technique is a key part in preparation for any exams, and it's something that you'll pick up the more you go and you do past papers. Right, so many questions in exams are repeats of what will be in old exams. So going and looking at lots of generic questions beforehand is a good idea. When going and answering and questions and reading them, there's a few things just to take into account. Make sure you go read the question at least twice so you don't make any mistakes when you're reading them. As you're reading them, go highlight key information. Don't make assumptions about what the question is asking. Make sure after you've answered it, you go check your work for daft errors that you might make. And if it involves going and answering something where there's some calculation, make sure that you write your workings out down. Right, let's go and have a look at the question.
Remember, an action potential is the part of the process that occurs when basically a neuron is firing. So during an action potential, part of the neuronal membrane is open or opens up to allow positive charged ions, right? It's important to say ions, not atoms, inside the cell and negatively charged ions out, right? But most importantly, it's the positive ions. So this process causes a rapid increase in the positive charge of the nerve fibre, right? So it's going and it's causing depolarization. So like we said, an action potential is a rapid change in the polarity that moves along the nerve fibre. So in order for a neuron to move from resting potential to an action potential, a short-term electrical charge needs to be established. So the level of stimulation that a neuron must receive in order to go and cause an action potential to be generated is called that threshold level or the threshold limit. Right? If it does not exceed this limit, then you don't get an action potential. So if we just go and have a look at this graph, right? So this graph is effectively showing our stimulus and the relative voltage of the neuron. So we can see just here, that is where we're going to have some sort of stimulus. The stimulus is going to go and cause sodium ion channels to open. Right, enough sodium ion channels need to go and open up in order to go and cause depolarization. Right, so it's that all or nothing concept. So if we go and we get depolarization happening, this is where the sodium ion channels have opened up and sodium ions are going and moving in through those channel proteins into the cell, right, causing depolarization. And it's that depolarization that goes and causes the action potential. So your depolarization is going and it's taking the voltage from minus 70 millivolts up to plus 40. Right, after that we go and we get repolarization where that refractory period is re-established. So we go and have a look at a quick question. So the amplitude and duration of action potentials in a single neuron are always the same. However, the amplitude and duration of action potentials can vary between different types of neurons, right? So that bit's important. So figure 2.2 shows the action potential in two different types of neurons. So we've got a Purkinje neuron and a dopamine neuron. So remember we're describing the differences between the two action potentials and you're suggesting explanation. So you're not just describing what happens. So we can see here that they are quite different, right? So some of the key things that you would need to go and talk about in terms of answering this question. So you can talk about both the dopamine neuron and the Purkinje neuron. So The dopamine neuron is less polarized, so you can see there that it only goes down to about -50, whereas Purkinje neuron goes down to about -70, -75, give or take. So depolarization shows less change because you're only going up to about +20, give or take, whereas in a Purkinje neuron it goes up to +30. So the action potential peak is lower, so you can see that that's lower there than it is there, for instance. There's no clear refractory period, so you can see here you've got your refractory period in your Purkinje fibre, whereas that's where it would be. There's no clear refractory period. So the action potential lasts a lot longer, right? So you can see, because it's going from kind of there to there really, where it's starting to vaguely repolarize, that is a lot longer than it is up here. So repolarization is considerably slower. Right, so that's if we were going and we were talking about the dopamine neuron. You can then go and talk about the fact that our resting potential is then set up, right? So we're setting it up by a number of different sodium and potassium pumps in the neural membrane are going to go and open up or start functioning. You can then talk about the fact that there's going to be a different number of potassium ion channels in the dopamine receptor in comparison to in the Purkinje neuron, so the dopamine neuron even, right? Which is why it's going to take longer, because it takes longer for that resting potential to be re-established. So there's a steeper sodium ion gradient in the Purkinje neuron, so the steeper potassium ion gradient in the dopamine neuron. So different types of neuron, or the different or more voltage gated sodium ion channels there's probably going to be, and the same with different numbers of potassium ion pumps. Right, so it's just about going and getting that detail in.
So hopefully you have found that useful. If you've got any questions, then drop it in the comments below, and we'll be uploading a lot more. Right, good luck, folks.